but now in chemistry, but now I think it's not important because I think we want to focus on applying the concept. So I think when it comes to polymerization and so on, it is more towards memorizing over here. Okay, so that is why less questions from this chapter. Yeah, less questions on this chapter. So let's actually, I will tell you what are the areas that we need to focus on. It's more towards bonding. Okay, you need to know more towards bonding. So basically in bio, you will learn that primary structure. Okay, is going to be known as a sequence of amino acid in the polypeptide chain. So it's just amino acid keep on one strand of amino acid and then keep on bonded to each other. So they are going to undergo polymerization, forming peptide bond. So each of them, okay, they will have the peptide bond over here. Okay, so it is written from N terminal. This is what they call it, N terminal, because the N, okay, it's actually written from the N terminal, okay, and then ends with the carboxylic acid terminal. Okay, why it is like that? Because each amino acids, they are going to have this carboxylic acid group, and they are also going to have the amine group, and then they will have H, and then this one can be the R group, right? That determines which type of amino acid they are. So here, if I combine them also, if I do another polymerization here, okay, with another amino acid, there will be this part, okay, is going to be at the end, okay, at the end. And then when I keep on do polymerization, there will be at the end, there will be NH2 over here. That's why it is going to be the N terminal. And do remember, this one, they can actually form the ion because something similar to zither ions. Okay, so the H plus will come over here. They will form NH3 plus over there. So this one is actually based on what we have learned. So this primary structure determine the ways it falls into secondary and tertiary structure. So we have total of 4 structures here: primary, secondary, tertiary, and quaternary. Okay, so the basic structure is the primary structure, and the way they are folded, they are going to be categorized into secondary or tertiary, and they are held together by covalent bonds. Okay, they are held together by covalent bonds. So this is actually between here and here. So that is nitrogen and they will have this peptide linkage, right? So this one, they are bonded with covalent bond. This is carbon and nitrogen is covalent bond. So all these are covalent. Okay, so here we have secondary structure. Okay, the next one, secondary structure is going to determine the areas of polypeptide chain which there is a particular arrangement of amino acid residues. Okay, and it is stabilized by hydrogen bonding, okay, between NH and CO group. Okay, and then this one, okay, more towards theory. Side chains are not involved. Okay, what are side chains? Let's say, for example, these are considered to be side chain. So they are not involving any side chain. We have 2 types of secondary structure. Okay, 2 types of secondary structure. So the secondary structures can be divided into alpha helix and beta plated as well. So it's going to be alpha helix and beta plated. They are going to be secondary structure. Okay, secondary structure. Let's actually focus more on here so that we can understand what they try to mention about here. Yeah. So if you look at this alpha helix over here, they say that it has a backbone that runs along the chain. Okay, so this is the backbone. Okay, that runs along the chain. And some points are flexible and allows free rotation. These are the points that are flexible, that allows free rotation. Okay, and here they already mentioned, yeah. So in the alpha helix, okay, the backbone twists around in so a rod-like structure is formed. Okay, why it can be twisted? Because there is a free rotation. Okay, you can see which these are the parts of free rotation, so they are flexible, so they can actually be twisted, and they are going to form some sort of a spiral form, and it is stabilized, okay, by hydrogen bonding between NH and CO group. Okay, later I'll show you the hydrogen bonding. So here, okay, you can see this and this, there is going to be hydrogen bonding. This and this, there is hydrogen bonding. So they are going to be this structure when they are twisted. Imagine they are twisted, and then there is going to be hydrogen bonding between them. So hydrogen bonding is between what and what? NH and then CO group. To be exact, hydrogen bonding is formed between this hydrogen and this O. Okay, so hydrogen and O. Yeah. So this is going to be the hydrogen bond lies parallel with the long axis of the helix. Okay, and then it's going to form a weak intermolecular link to CO group. Now, side chain residues stick out on the outside of the helix. Okay, so this, if you look at this, okay, you can see that side chain, they are going to stick out like something like this. This is R. This is going to be the side chain. Okay, I'll show you the 3D structure. Yeah. So this is the R group. This is the R group. They are sticking out. Okay, they are sticking out, and the R group, okay, the R group does not involve in the hydrogen bonding. So if you refer back to what I told you earlier, okay, it is stabilized by the hydrogen bonding between this group. Side chains are not involved, okay, when it comes to hydrogen bonding. Okay, so this is going to be what I explained to you earlier. So the side chain just stay there. Okay, they just stay there. Okay, they don't get involved. The spiral, okay, the spiral that they are going to form, it's going to be balanced by the hydrogen bonding. Okay, it's going to be balanced over there. Now, when we have this beta pleated, okay, beta pleated, this is going to be secondary. Hold on, yeah. Now, when we look at this beta pleated, okay, secondary also, okay, so you see this one alpha helix is also secondary structure. Okay, we can see now, I think if you want to compare, what is the difference between primary and secondary? Secondary, they will start to have shapes, okay, 3D arrangement, okay. So they are going to be having some sort of 3D arrangement. They are going to have hydrogen bonding, okay, because previously it's just, this one is just covalent bonding, one strand of polypeptide chain. So that is primary. But now this primary structure, when they start to be arranged, you can form either alpha helix or you can form the beta plated. Now the hydrogen bonds are formed, okay, again similar NH and CO. So they have actually mentioned it, they put it in this way so that easier to see. So this is the hydrogen bonding. So the hydrogen bonding will be there. Okay, so of the different polypeptide chain, or okay, different areas of the same polypeptide chain. So basically, if you look at this, okay, there is like something like a your graphite, like something like layers. Okay, there are layers, and the layers are connected or stabilized, okay, by this hydrogen bonding. Okay, so the layers are being stabilized over there by hydrogen bonding. So from where to where? So it's actually bonded from here to here. So between NH and CO, and they will maintain, okay, the beta plated structure. Okay, this one is one of the structures found in silk. Okay, so it is going to be, again, you see, one of the things that we can compare between primary and secondary is Primary will have only covalent bond, secondary will have hydrogen bond. Okay, if you have beta plated, they have layers. Okay, if you have alpha, they have helix, something like a single strand DNA. Yeah, not actually DNA, they are combined with two strands, right? So this one is going to be single strand. Okay, so alpha helix. Now, what if you look at this secondary structure? So basically secondary structure, okay, they have combination of both alpha and beta. Okay, so if you look at here, pepsin, okay, this is going to be an enzyme, okay, in your stomach. Yeah, pepsin has alpha helical region and beta plated region as well. So beta plated, they actually show in form of arrows. This is beta plated, and then here all these are going to be the rod light is going to be the alpha. So it means the combination of this, okay, the combination of this structure forms the structure of this enzyme. And the way that they are arranged, okay, remember last time we learned about catalyst, and enzyme is a biological catalyst. So how enzyme is can function as a biological catalyst by having more specific structure, where they will actually have this lock and key model. So they are specific because they will form some sort of a structure so that the substrate, they can actually react using the lock and key model. Okay, so it's very specific. So biological catalyst is more specific than your inorganic catalyst, like iron catalyst and so on. So that is the reason how they work. Okay, basically by the arrangement, okay, of the secondary structure. So enzymes, okay, will have secondary structure. If you move on, so let's actually look at tertiary structure. Okay, tertiary structure, they involve further folding of the polypeptide chain. Okay, there will be more folding. This one is okay lah, the folding is still level 1, but this one is going to be more folding. It involves further folding. The complex structure is stabilized by not only hydrogen bond, yeah, not only hydrogen bond, they are going to be stabilized by weak van der Waals forces, ionic bond, okay, which is salt bridge. They are also being stabilized by disulfide bridges as well. So if you look at this, okay, this is going to be one example. Okay, you see this is going to be hydrogen bond. Okay, so now you can see earlier I mentioned about in secondary structure, the R group, the side chain, they do not involve in bonding. They actually stick out. Okay, but now all these are side chains. Okay, they are involved in bonding. Okay, the side chains are involved in bonding now. And these side chains, you can see they will form hydrogen bond. Okay, if they can actually have these disulfide bridges. Okay, sometimes if you have something like this, okay, you know the one end of this primary, you can see here. The primary, you have this end and this end. What happens if you connect them together? That is possible also, negative and positive. Okay, so what happens, this thing will happen only in tertiary. So you can see that they connect the ends as well, positive and negative. It's possible. This is ionic bond, and so, this is going to be the side chain and side chain. Okay, the side chain and side chain, they will give you van der Waals forces of attraction. So there's so many attraction that is going to happen in this particular tertiary structure. So the structure is going to be stable, and they will have a complex structure over there. And yeah, basically this one is going to be the diagram to illustrate the nature of interaction and the bond stabilizing the tertiary structure. Okay, so if you try to recall again, yeah, primary covalent, secondary, they have hydrogen bond only. But tertiary now, they are going to have disulfide bridge, sulfur and sulfur, okay, van der Waals forces, okay, hydrogen bond, and ionic bond as well. So there are total 4 bonds that is going to be. So if you have more bonds, it is going to be, we can say that this is more stable or more stronger compared to secondary structure. So imagine that this is going to be protein. Okay, it's more complex protein. So that's the reason why sometimes I think in bio you learn that when you have higher temperature or different extreme condition of pH, your enzyme will become denatured. Okay, so the enzyme, the bonds will be broken. So what kind of bonds that are actually being broken over there? It's only the hydrogen bond. Okay, the hydrogen bond is being broken, but does it mean that suddenly if I go and go into a very high temperature, okay, and then uh, all my protein will become denatured? It's not going to happen. Maybe enzymes, okay, cannot function properly, but protein, okay, they do actually maintain the structure. Okay, they don't actually suddenly become denatured like enzyme. Okay, they don't lose their function, they don't lose the shape. Okay, because of the other bonds that are going to be involved over here. But if you look at the strong bonds, even the hydrogen bond they say is weak. Okay, the strong bonds that we are talking about will be more on the disulfide bridges and ionic bond. Those are the two bonds that will be really maintaining the structure of tertiary structure. So if I move on, okay, you can see here disulfide bridges, the strong bonds here. So they are usually found in proteins that function outside of the body. Okay, outside of the body. Okay, so this is going to be digestive enzymes. Okay, are usually disulfide bridges are usually found in proteins that function outside of the body. Let me check on this here, whether it's outside or inside the body, because digestive enzymes, they are inside the body. Okay, so anyway, maybe I wrongly typed this. Okay, but just double check. But okay, disulfide bridges are going to be found in digestive enzymes. Yeah, so disulfide bridges can be formed within the same polypeptide chain or between different polypeptide chain. Okay, they help to maintain the tertiary structure by locking them in place. Van der Waals forces is when non-polar amino acids are close to each other. It plays in stabilization. Hydrogen bond between polar side chains. Okay, ionic bonds are also formed between positive and negative ion. So basically, just to tell you back about all this that I mentioned just now. Okay, you can see some structures. All these are taken from your textbook. Okay, so if you have a cysteine, okay, how do you get cysteine? A different amino acid, they have different name because of the different R group. So the R group for cysteine, they will end with CH2 and SH. So if you have cysteine, another CH2 and SH, cysteine residues, the side chain, they will interact with each other. How? By forming the disulfide bridge. Okay, so in when you're forming the disulfide bridge, okay, the hydrogen is gone. Okay, so if you have, let's say for example, hydrogen bond, so if you have this side chain and you have this side chain, you can see the oxygen and the hydrogen, they can form hydrogen bond. Okay, so this is going to be between serine and uh, threonine residues. Okay, and this one, okay, you do not need to know what residue and so on, but if you can see the structure, you should be able to tell what kind of bond will be formed. Okay, this one is going to be positive and negative. Of course, it's going to be ionic bond. Okay, so this is going to be ionic bond. Okay, so all these ionic bond, hydrogen bond, van der Waals forces of attraction, disulfide bridges, they are going to maintain tertiary structure of protein. Now, this one, hydrolysis of protein, is similar to your hydrolysis of amine. Okay, this is actually when you want to break this part. Okay, but okay, we have learned about, remember when we learned about this the reaction between, if you want to break the bond, okay, let me show the previous. This is what we have learned earlier. Hydrolysis of a substituted amide. Okay, if you look at the substituted amide, okay, if I want to do hydrolysis, I try to break this, yeah, amide. When I try to break this, I am going to get RC double bond OH, and I am going to get N based in this example R2, then this one will become H. Yeah, so you will get carboxylic acid and amine. And then depends on what we use, they will further react. Like for example, if you are using sodium hydroxide, then you will get the salt of this. Okay, if you are using acid, okay, then this if you have acid, then they are going to actually have this R2NH3. It means that the H plus will react with the base. Okay, so all this will happen. So similar reaction is going to be happening over here as well, because the structure, okay, of protein, they are the same. Okay, so if they are same, so we can actually do this acid reflux or alkali reflux, and the product is almost going to be the same. So you can see here, I've written that, okay, if you do the products of hydrolysis are going to be the original carboxylic acid. Okay, it means in this case you are going to get the original amino acid. Okay, now once you get the amino acid, let's say you do the hydrolysis. Now let's say for example, if I do hydrolysis, I'm getting the amino acid. Like for example, I have amino acid is you are going to get carbon, let's say this one, this part, and then you will have NH2, and then one R group, and then H. Okay, this is going to be the amino acid structure, the simple amino acid structure. You are going to get this. But if you are doing the hydrolysis, okay, if you're doing the hydrolysis using HCl, okay, what is going to happen? Okay, the HCl is acid, so this H plus will come over here. You are going to get NH3 plus. Okay, if you are using sodium hydroxide, this will stay. Okay, but this part, okay, this part is going to become O minus and then Na plus. Okay, similar like what we have learned earlier in the previous chapter. So this is what they say, the amine group, they will react with the excess acid, okay, to form the ammonium salt. Okay, so like this is going to be formed. Okay, so if you are going to get the amino acid, okay, but if you are using acid, acid will react with the NH2 part, they will give you this one. Okay, with excess alkali, okay, the NaOH, you are going to get the amino acid, but you are going to get this one because the carboxylic acid group will further react with the NaOH to give you this ammonium salt. Okay, so similar. Okay, the only thing is reflux. Okay, so when you do reflux of protein, okay, it is almost the same as what we have learned in your amide. Okay, for amide, you are going to get one carboxylic acid, another one is going to be amine. Then we do, but in this case, both of them are together. Okay, because this is going to be amino acid. So in that particular molecule, one part will react, another part will not react, depending on what you use. Okay, so this is going to be the questions over here. Okay, let me see here. If we look at some of the questions over here, okay, we have this. I think this one is done earlier in IGCSE. What is addition polymerization, and what is substitution polymerization? What is addition polymerization and what is condensation polymerization? Addition polymerization is going to be a polymerization whereby, okay, you are going to add, okay, it involves the double bond. Okay, so you are going to involve double bond. Like this is one example. of addition polymerization. If the monomer is having double bond, okay, so what happens? This one you break into two, okay, so what happens if I draw, okay, this part, half of it will come here, another half will come here, so this one will be gone. So this is what you are going to get, okay, you'll get this and this, okay, this hand will extend here, this hand will extend here. This is going to be involving monomers that are unsaturated. Okay, unsaturated means anything that has double bond, they will undergo addition polymerization. So what is going to be condensation polymerization? Is quite simple. If you can react like ester, polyester formation. Okay, ester, you get it from carboxylic acid. So carboxylic acid. And then if you have alcohol, okay, we know that the H will come here, OH will come here. You are going to get RC double bond O, then O, R2. Okay, if this one continues, okay, this is a simple ester. Okay, ester. What happens if I want to have polyester? So in order to get polyester, you do like this: C double bond O, OH, something in the middle, but they can have C double bond O, OH here. Then only you can have ester, polyester. And then for alcohol also, they are going to have OH here. So something bonded, okay, then another OH here. Then only the reaction can continue. Like for example, if here the OH and the H, you are going to have the ester linkage. The reaction will continue with another part over here. And the reaction will continue with another part over here. This is what we call polyester. Now, this is polyester, and I can say that this is a condensation reaction because they are removing water. Every single time one linkage that is being formed, they will give you water. So this is going to be example of condensation polymerization. Now, so you see which one is addition. You see the moment you see there is a possibilities of having double bond here, addition. This one will be condensation. This one will be condensation. Why? Because carboxylic acid will react with this part, another part of amine. So they are going to actually give you the polypeptide. Okay. So this one also is going to be also condensation as well, because carboxylic acid can also react with alcohol. So explain the basis. You can see addition polymers are something that has alkenes. Condensation have monomers. They are not alkenes but have two functional group per molecule. Okay, two functional group per molecule. Here I have one example here. This is going to be two same groups. Okay, but here for protein, they have two different groups in one molecule. So you take note as well. That's the difference. And then write an equation between propene molecules. Okay, they already write, okay, the equation. And then what type of polymerization? Addition polymerization. What is the repeating unit? Okay, the repeating unit is only this. Okay, so this is the repeating unit. I have given one example just now related to this, but I use OH. But now if you have amine group left and right, also possible. Okay, so please actually go through all this. Okay, this question I have already given the answers also. Okay, please go through that. And then please also go through this naming, disulfide, what is amino acid residue. Okay, all this has been done. I'm very sure you can also do this. Okay, all this has been done. What part disulfide bridges play, okay, in stabilizing? Okay, so basically they are going to actually bridge across different parts to keep them in the correct position. Okay, so they are quite strong. Okay, so it is stabilized. Okay, please read. So now, Okay, the last part over here, we are going to look into DNA. So I told you we can actually definitely finish the chapter today. Yeah, there's a lot of things that here no need to focus so much. Okay, even DNA, they used to ask a lot of questions, but now no more. Okay, not a lot. Okay, so DNA is deoxyribonucleic acid, two functions. Okay, they make copies itself so that it can be passed, okay, through generations, and it contains sequence of bases, okay, that form the genetic code to synthesize protein. So you see, all like bio. Yeah, so do we need to memorize this? Okay, this one no, but here the component, okay, the component of DNA you need to know. There is a sugar, okay, deoxyribose, okay, it is 5-membered ring. So this is the sugar, and then they have a base group. Okay, the base is going to be a nitrogen containing group. Okay, so here now this base have 4 types. Okay, I think those bio students you will know. Okay, A, T, C, G, and so on: adenine, thymine, cysteine, guanine. I think I forgot already all this. Yeah, so these are the thing, and then there is a phosphate group. Okay, the P group. So the phosphate group, so you can see if you just write down everything properly, this is phosphate group. This is the R group that has the base. Okay, this is the base. So this is going to be one basic component of a DNA. So here, I think more towards bio, yeah. So you see, A always pair with T. Okay, G always pair with C. Okay, all these are bases. Okay, when it comes to DNA. So adenine and guanine have planar structures with 2 rings. Thymine and cysteine have planar structure with single ring. Okay, DNA consists of double helix. Okay, 2 strands which keep in place by hydrogen bonding. And you can see over there, I think all these bio parts I don't want to actually go through. Okay, please read. Okay, so the nitrogen containing bases, okay, this is adenine, guanine, thymine, cytosine. And these are the hydrogen bonds. Okay, so A always pair with T, C always pair with G. So different combination that we have. Okay, because I think we have 20 amino acids. If you pair them in different combination, and they are in this case 4, yeah, all these 4, they will have different configuration, different arrangement, different sequence. So they will create a very unique structure of DNA that is actually unique for every individual. Okay, so that is going to be DNA. So base, okay, DNA base on one strand linked to another particular base on the other strand. They form complementary base pairs. I think it's bio, all these terms. Okay, the structure is kept stable by hydrogen bonds, van der Waals forces. Hydrogen bond is not regular. They are not in line with each other, twisted, and this makes the DNA double helix to bend rather than being a straight rod-like structure. So they are able to rotate. Okay, so done. Okay, any questions?